And can you all see my screen okay? Or the slides?
Yes, it looks great, Jen.
Thank you. All right, so we're really happy to have you all here today in the ESS-DIVE community webinar. My name is Joan Damerow, and I'm the community engagement lead scientist for ESS-DIVE. And on May 24th and 25th, we actually held our first ESS-DIVE community data workshop. And in this workshop, we introduced ESS-DIVE features and goals, and around 80 people came. And this included a variety of researchers within the environmental system science community. And we had tutorials on how to use ESS-DIVE for data management and publications, and a number of community discussions. And one major takeaway from the workshop was that people really seemed to value the tutorials. And so what we're going to be doing in some of our webinars now is to provide more tutorials throughout the year. And for this webinar, I'll be leading you through an interactive tutorial on how to use ESS-DIVE's sample identifier and metadata reporting format. And we developed this reporting format over a couple of years, actually, in collaboration with many members of the ESS-DIVE community. And we've had several webinars in the past to introduce issues around sample tracking, and also to discuss the metadata, the sample identifier and metadata reporting format that we developed. But today is going to be more of a tutorial format. Before we get started, I also have a few poll questions that we can use to all learn a little bit more about who's here today. So you can visit the link provided in the chat by Madison to access the first question, which is, what is your primary role in your research group? We have a lot of data managers here today so far. I'll just give you a little bit more time here. If you do have others, maybe put in the chat what that is, what your primary role is. That would be great to know. Okay, so it looks like most of the attendees today are data managers, but we also have some field scientists or data collectors and some lab scientists and PIs. So we're really happy that you've all joined today. And I'll move on to the next question. What organization are you representing today? And this is a free text question, so you can enter KBase, LVO, NMDC, NG Arctic.
Do we just type this in the chat here?
So there's a link in the chat for a poll.
Oh, I got it.
Everywhere.
I got it. Yeah, okay.
A couple people from NG Arctic, University of Alaska. University of Wisconsin. Okay, great. It looks like we have a few different organizations represented. Sorry. Okay, so which of the following terms are you familiar with? We found in some of our previous webinars that not everyone was familiar with some of the terms. And so if you have attended some of our past webinars on this topic, or if you attended the workshop, some of you may be more familiar with some of these because we discussed them previously, but many of you may not as well. Okay, so most people are familiar with, well, more people are familiar with metadata and dataset. Unique identifiers, and then less so persistent identifiers and IGSNs. And we will definitely be discussing definitions for these terms in the tutorial today. So I'm going to go ahead and advance to the next question. What are you most interested in learning about today? How to assign unique identifiers to improve sample management and tracking, when you may need to register samples for IGSNs, and how to make sample data more discoverable and usable. Yeah, so a lot of people want some more information on how to assign identifiers and when to use IGSNs in particular. Just a couple seconds here, and then it looks like the responses are slowing down. Okay, great. This is great because these first two will really be focusing on today in the tutorial: how to assign unique identifiers and when you may need to use IGSNs and how to do it. So throughout the tutorial, this is the plan. We're going to first discuss sample identifiers and metadata and what some of these terms mean. We will introduce the ESS-DIVE sample ID and metadata guide and template, so you know exactly where to look for information in the future. We'll then go through some of the specific workflows on how to plan, manage, and publish your sample data, and how and why you may want to use IGSNs. And so this link will take you to a Google Drive folder that contains several files that will be useful in the tutorial. And you can reference this later as well. And so please let us know actually if you have any trouble accessing the folder or individual files, and if someone can monitor chat, someone from the ESS-DIVE team, and let me know, that would be great. One of the files is a shared notes document, and this also contains useful links that are referred to throughout the presentation slides. So you could open that as well, or just monitor the chat if you want to visit the links that we're discussing throughout the presentation. And the takeaways that I hope that you'll get from the tutorial are first, I hope that everyone will understand more fully when to use unique identifiers and persistent identifiers and what they are. Learn about what it looks like to standardize your sample metadata using our spreadsheet template, how to register samples for IGSNs when you need that for your project, and then how to publish your sample data sets on ESS-DIVE when all of your analyses are completed and you're ready to do that. We have done research to develop guidance for sample identifiers and metadata. And the purpose of this is to enable more effective sample planning and tracking, and also discovery and reuse, so that you can maximize the value of your sample data over time. So it's both about helping you with your sample management, and then also helping preserve that data so that it's more discoverable and reusable. And here's a little terminology check. We had the poll about these some of these different terms, but we can review what those are before we get started here. So identifiers are really essential to document and track metadata over time for your project, and also to link related information across your files. So these concepts are really important for thinking about what identifiers you need for your projects for things like samples. So a unique identifier provides a meaningful project-specific unique ID. Unique identifiers for things like samples and site locations are kind of like your name. Your name is meaningful for your friends and family and coworkers, but it can also change over time. Hopefully, maybe it does or doesn't, but it often does. And you can have variations on your name in different locations. And many people actually have the same name as you. So when that is presented in an online system, it becomes more complicated to track that. Persistent identifiers, on the other hand, are globally unique IDs, and they include a permanent link or a landing page and associated information about what that identifier is referring to. And so these are kind of like your social security number. They're not really meaningful to your friends and family in most cases, but it doesn't change over time. And this is how important information about you can be tracked. Metadata is then the descriptive information about data. Metadata provides useful information to describe samples, such as the sample type, the environment it was collected from or feature, and the location. If you think about it in terms of yourself in this analogy of your name and your social security number, the metadata is something that can help a recruiter find you, for example, or it can link you to relevant music and products that you're likely to enjoy. So hopefully that will help you distinguish these different identifiers and when you might want to use them. So what is sample metadata specifically? This is the information that characterizes samples and where, when, how, and why they were collected. It includes details such as sample collection details, like the collection date and the time, the collector, chief scientist, the collection method description. Also, it includes information about the location, the latitude and longitude, a location description. It includes the environmental context is often very important for physical samples, the local environment or physiographic feature from which it was collected, and the broader ecosystem or biome. Also, there are a number of fields that characterize the sample material and the sample itself, such as the material, some more specific classifications, the purpose of sample collection, etc. One other key piece of metadata that you might not always record or think about is recording related identifiers. And this can be particularly important for multidisciplinary projects. And it's how you can track relationships between related samples and other entities like locations. So in our sample reporting format, the related identifiers that we have are parent IGSNs, so you have parent-child relationships, and we'll discuss that more later. Also, broader collection IDs, event IDs, and location IDs. And I'll discuss these in a minute. So in some cases, a locally unique and meaningful project sample name may be sufficient for analyzing and tracking samples within a project. So when might you need a persistent identifier for samples, like an IGSN? And these are just some cases where IGSNs are most useful. And this includes projects where samples might be used in multiple data sets or journal publications, where the samples may be sent to multiple collaborators who work on the same samples. They're sent to multiple labs for a variety of different analyses. Sample-related data may exist in numerous different repositories or more than one repository, or they may be just used over time for multiple purposes. So I'll just take a minute here to ask if there are any questions at this point. And if not, I'll continue, and there'll be plenty of time for questions throughout, and feel free to ask questions in chat or unmute and ask them throughout the presentation, because this is an interactive tutorial.
I have a question.
Yeah, go ahead.
Yeah, I put it in the chat, Joan. Thank you. When do you not need persistent identifiers? I mean, it seems like from those 5 things you listed, it seems like anything you do might be published. It might have a collaborator. Like when is it sort of safe to say, like, I really don't think I'm going to need persistent ID?
Yeah, I think it just depends on the extent to which you're working with different people and labs on sample data. Like some sample data, it may be like very short-lived, and you just analyze a sample for a certain analyses, and that's all you do. And you're going to publish that data, but it's probably not going to be in multiple data sets.
Okay, thank you.
Yeah, does that make sense? I mean, it seems like within the environmental system science community, I mean, we were having, we had a lot of feedback that sample tracking was a big issue for some of the multidisciplinary projects. Particularly, a lot of these projects are do have multiple collaborators, and that was creating some issues with sample tracking. And so that's why we started working on this reporting format and recommending IGSNs in particular. Okay, so persistent identifiers, again, like IGSNs and DOIs, they have web landing pages. And these pages have both machine readable and human readable metadata descriptions about the sample or data set. And this includes things like the title and abstract for data sets, and the sample type and collection locations for samples. And it can also include links to related metadata and data. That's what's shown down here on the bottom of the IGSN landing page on the left. So we have links to a number of different other related resources, including 2 ESS-DIVE data sets. And on the right, we have an example of a related ESS-DIVE data set. And so you can add links to related data sets on your sample landing pages. And then you can also add links to the original samples on your ESS-DIVE data set. So why would you want to add links to related samples and data? Samples provide a good example for how multidisciplinary and interdisciplinary data in particular may exist or originate in different data systems. So for a given project, FTICR analyses may come from the Environmental Molecular Sciences Laboratory, or EMSL. Genomic sequencing data are published on JGI's Genomes Online Database, or GOLD. And modeling work may take place on the DOE Systems Knowledge Base, known as KBase. So the persistent identifier can provide a really powerful way to link and exchange information across these facilities and data systems. And in turn, this can improve our ability for people to find, compile, interpret, and reuse this multidisciplinary sample data. Otherwise, it can be very disconnected and difficult to compile all of the related data. So we've just begun working through some use cases to add URL links with descriptions to test how we can associate the CSAR IGSN landing page and the ESS-DIVE data sets in a meaningful way, also to associated data in GOLD and other data systems like the National Microbiome Data Collaborative, KBase, and EMSL. So our goal in this work and coordinating across these different data systems that many people in the ESS community are using is to determine how we can add related links automatically in the future. We aren't able to do that currently, but we're working on plans to see what we might be able to do to add this information automatically to make publishing interdisciplinary sample data easier and make the published data more complete and usable. So this kind of gets at the last question. Which of the following applies for your project sample data? Just want to get an idea of where people are with this, with their projects. So these are the different criteria that I mentioned earlier on when you might want to consider using IGSN. So if you're selecting any of these, then the answer is probably that IGSNs, you should consider IGSNs for sample tracking. So it looks like the most common is that people are working in teams where multiple collaborators work on the same samples, or that the samples are included in multiple data sets or journal publications. Also, that sample data is stored in different data systems or sent to multiple labs for analyses. And there are just a few where none of these apply. And this tutorial is still relevant. If none of them apply and you don't think IGSNs are going to be useful for your project, you can still use our sample metadata template to help make your sample data more findable and usable. And also using the project-specific unique identifiers to help with your sample tracking. So now we can get into some of the specific steps that you can take for sample planning to publication workflows. And so this includes planning your sample campaign, standardizing sample metadata using our spreadsheet template, registering samples for IGSNs when you've found that this would be useful for your project data management. And then finally submitting your sample data sets to ESS-DIVE when your analyses have been completed and you're ready to do that. So step one is planning, where you can determine your file organization and identifiers that can help organize your sampling campaign and the subsequent data that you're going to collect. For many projects, a lot of your samples may turn out to have the same metadata. For example, you may have a collection of samples that have a common purpose and they're all to support a specific project. So you can provide things like purpose and project name and chief scientist and country. You could provide that in a higher level file to describe a collection of samples. That's a little bit less common. The most common is to provide a metadata for all of your site locations in a locations file. This may include just all of the information about your locations, including a location ID, and then also the descriptions of each location, like latitude and longitude, and a location description. The most important identifier to consider is the sample name. This is this is the project-specific, meaningful unique sample name for all of your individual samples. And this is because it's required for using the samples reporting format, and the other identifiers are optional. So in general, this diagram is just demonstrating how you can separate metadata into different files to describe higher level like collections of samples, locations, or sampling events. And the purpose is just so that you can provide this information efficiently and not have to provide that same metadata over and over again for each individual sample. So in general, with planning, you can start by determining your approach for assigning project-specific unique identifiers to describe things like locations, samples, and child samples or subsamples when you have sampling hierarchies. And again, a common and efficient way to do this is to have some separate files for things like locations and then another file for all of your sample metadata. And when you use identifiers in this way, you don't have to provide the same redundant location information, for example, for all your samples collected at that location. You can use the location ID to track that information and then just provide that location and ID for all the samples that were collected at that location. The reason that we went this route is because we had feedback that's preferable for many projects that prefer to manage metadata in this way. You should consistently use your unique identifiers to manage and link related metadata and data across files. For example, this means that you need to consistently use the same unique sample name across all of your files that contain relevant sample data. It does happen in many cases that people change these identifiers, and it creates sample management issues down the line. And here's an example of how you might separate metadata into 3 different files: a locations file, a parent sample file, and a subsample file. And you can see that the first column of the locations file contains all of the location IDs, and these are all unique. And the subsequent columns then provide all of the location metadata, including things like lat long and descriptions of the sites, etc. And then the parent sample file contains all of the specific metadata to describe the samples. It also includes the location IDs for that location, for the location that each sample was collected from. And so this is one particular location ID that is used across these 3 different files. The child sample file then also provides the parent sample or parent IGSN to link those child samples to the higher level parent sample. And the parent sample or parent IGSN is outlined in blue here. So now we're just going to do a short activity where you can think about how you might organize your samples. And so you can use the link provided in chat to access this, and it will take you to this file, the sample relationships activity. And what I want you to do is to claim a slide. Just go to a slide and fill it out, and you'll be basically just completing an example of one of your own sample hierarchies. And to do this, you can modify the boxes as needed and fill them in with your sample type and the unique sample name that you might use for that sample or location. You can also consider using identifiers for a site or location. And when you go to one of the slides, there's a little description of what to do in the box at the bottom. And then also, it'd be great if you can enter your project name in that text box on the left and the purpose of your sample collection. And what I'm hoping that this activity will do is help you think about how to link related samples. Maybe you do this already for your project using the location identifiers and the sample names, your project-specific unique sample names. And then also for us to see what some of your use cases are. So we'll just take a couple of minutes and let you all fill this out. So it might be quiet for a couple of minutes, but also you can ask any questions. This would be a good time to ask questions about this or have a little discussion about it. So if you do have a question, you can speak up and unmute, or you can enter a question in the chat, or you can simply just fill out your slide. Often the sample name tends to include just certain details that are codes that enable people to quickly understand what that sample is, such as a code for the sample type and the location and the date. That's a common way that people do that. And that many of you may have done already in the past. We'll just take another minute or two. It looks like people are still working. You can also revisit this later as you're planning your samples work and thinking about how to use identifiers. And we have links to this as well as a more involved sample journey map as tools to help with this task of assigning identifiers and the project specific sample name and the IGSNs. Yeah, it's really great to see some of these examples. And if anyone has questions on assigning identifiers, you can ask them now, or you can always contact me or the ESS-DIVE support at a later time, and we can schedule a meeting with you to go through some of this for your specific project, because for many projects, this can get pretty complex when you start going through the complete sampling hierarchy for some of these collaborative interdisciplinary projects. So I think I'm going to, does anyone need more time right now, or are we ready to move on? Or does anyone have a use case that they want to discuss?
Well, yeah, this is Eric Roden from UW-Madison. I'm like totally green in this.
Mm-hmm.
I have not done this before. And that's why I'm here. I mean, the idea, I am still just trying to get my mind around the overall structure here. So these sample IDs are that at one level or another, they're hierarchical. So that then you can link all sorts of different, I was working on slide number 3 there. So that you can link all kinds of different experiments to a given project and sort through. And like, what's daunting about this for me, and you probably hear this from other, semi-Luddite people that have never done this, is, we keep good records, but we don't like assign sample IDs that are, searchable and interrelatable for different projects. But that's what we're trying to do here, right? And let me just make this real, and I'm sorry for hogging time, but like, what I entered there is for an in situ experiment that we're hoping to conduct this summer in August at Hanford at the 300 area location in the river.
Mm-hmm.
So location ID and sample name. Do I want descriptive names in those boxes, or am I supposed to have, would those descriptive names go somewhere else in the database?
So this activity is specifically for your project-specific unique identifiers that help you track information. So often those are fairly descriptive, but there's another field that you can use for a longer description. There's a description field where you can really fully describe your sample or your location and whatever. It's free text, it's open, so you can describe it however you need to for your project. But these sample names and location IDs are usually, fairly short so that it's, fits in a cell and it's easy to use to track information. And so the location IDs often have some like shorthand code for your site, like Rock Creek, like ROCKCR kind of thing, and then the date. Well, that would be more like a sampling event. But yeah, so it's however you want to format it for your specific project. And then again, the sample name often includes some code for the site and the sample type and the date, but you can do it however you want. And often some of the sample names that you probably already use would be great to include here if you are using unique sample names.
Yeah, but what I have there, as unintelligible as it may be to you, is not too far off.
Yeah, exactly. And you can use this, and then you can include more info. Like all of the contextual information is really going to go in your other sample metadata fields, as long as this works in your project and it's a unique identifier that your project consistently uses to track that information. Then that's great.
Yeah, the wacky thing is we've never done this before, ever.
Yeah, yeah. A lot of people are.
It's like so different. We keep all kinds of notebooks and all kinds of records, and we know what we're doing, but we've never done things like this. So hence my kind of fumbling questions.
Yeah.
But thank you. This is great. This is exactly what we need.
Yeah. We are starting to run a little low on time, so I think we need to move on. I think Donnie has a question. Do you have a quick question that we can address fairly quickly?
No, it was actually a comment to address Eric's question. I'll just do that in
Okay.
the chat. It's not an additional chat question.
Okay, great. So I'm going to move on so we can get through the rest of the tutorial here. So the next thing is just to access our guide. If you access our documentation for samples, again, I mentioned this before, but this contains all of the information that you should need for using our reporting format. And it also includes information on how to contact us for more questions or more information. Here's what it looks like. We're using this tool Gitbook for our documentation. So all of this is also mirrored in GitHub if you prefer to use GitHub. So on this page, it has a description, also getting started. There's a link to our metadata reference guide and an instructions document. So I'm going to click on this on how to assign sample identifiers and register samples for IGSNs. And this includes a lot of the information that we're discussing today in the tutorial, like file organization, sample hierarchies, standardizing your metadata, et cetera. So you can scroll through here and for all of the steps that you need to use this. We also have this IGSN metadata reference guide. And at the top of this guide is a list of all of the sample metadata fields. And when you click on a given field, like material, it'll take you to a small table that describes what that field is, including the definition and the format that we're asking for. There are a few fields that require using a controlled vocabulary. It's just a few, including material and the object type and the physiographic feature. And here is the materials terms that we have for ESS-DIVE. And I'm going to visit this again in a minute. So we also have a paper that describes our work on samples. And the DOI is provided here. Again, this was based on some extensive research on existing standards and also a pilot study where we worked with 8 different national lab projects to actually go through the process of standardizing their metadata and assigning IGSNs. And so if you want to learn more about what we did there, you can visit and read our paper. So the next step is providing your standard sample metadata to characterize all of your samples and your collection details. And you can download the template. Let me show you also, you can easily access the template from our documentation. In the Getting Started section, there's a link for the sample metadata template. And then you can go here and then click on download. But this template is also in the folder that we shared at the beginning. Here, the sample template. There's just a couple of different ways that you can access the template. And it's just a spreadsheet. There's one header row where you provide the object type. In many cases, that's going to be individual sample and then a user code. And I'll discuss that more in a minute.
Joan, there's a question in the chat from Les about whether it's better to start by completing the IGSN metadata or the ESS-DIVE template.
It's only the ESS-DIVE template. The ESS-DIVE template provides all of the information that you need for registering samples through CSAR. So don't use the CSAR template. You should use the ESS-DIVE template that's provided in our documentation. And all of the metadata that we have in our reporting format is based on CSAR IGSN requirements. There are just a few additional fields that we found were needed for many in the community to more fully describe some of their samples. That included things like adding the higher level IDs, like the location IDs. And we also added some more material terms. And we worked with CSAR actually to add some additional ones. So if you use our template, you'll be all set to register samples for IGSNs. So I just wanted to discuss these material terms quickly. and go through a poll question potentially. So let's see, this is the link to our materials terms again. Currently, we're using the CSAR IGSN template, and they have a number of very high level material terms to describe samples, and they're all listed here. There are a few cases where we found that we needed some more specific terms, but we still need to use these higher level material terms to interoperate with the CSAR IGSN metadata guidelines. And so we're using these higher level terms. And then if you have a more specific material classification that you need or want to use, you can use those by providing them in a field name informal classification field. And so that's just for water samples in particular. We have a number of different categories to indicate surface water, groundwater, and pore water. And so here's just a quick poll to gather some information on what your sample materials are.
Joan, there's a couple questions in the chat you might want to look at while people are filling the poll.
Thanks. Yeah, okay. So Donnie has a great question. The template has only one sheet. I thought there would be sheets for collections, locations, methods, events, samples, and subsamples in the slides. Are there templates for that approach? So basically, you can provide all of your metadata in one file. It's just an option to have it be provided in different files if you don't want to have to provide that same redundant metadata. It's the same metadata fields. So you just create a new file for your locations and just use only the metadata that's relevant for those locations, and you can delete all of the other fields. So it's basically the same template. It's just providing that information in different files if it helps your project data management. But it's optional. You can provide it all in one file. And then let's see, are there any others? Okay,
There's
so
a question from Stephanie as well.
okay, so in the template, is it one metadata file per sample, or can I have multiple rows? Oh yeah, definitely. It's you definitely should provide multiple rows to describe all of your samples. You do have to have a new file for if you have different object types. For example, you would need a file, like if you have a series of soil cores, like say you have a soil core, and then you want to describe all of your core sections. That would be one file. And then if you have individual samples that are like subsamples from those core sections, you could provide that in a separate file, and those would be individual samples as an object type. Yes, so yeah, soil cores would then be separate from the tree samples. Okay, so most people have soil samples. I'm gonna move on to get through some of the rest of the tutorial. Step 3 is then registering your samples for IGSNs. So again, if you send samples to multiple collaborators or different labs for a variety of analyses, or your data is going to be published in different data sets or publications, you would likely benefit from using IGSNs. And step 3 is registering your samples for IGSNs using the System for Earth Sample Registration, or CSAR. So to do this, you choose a 3-character user code for your project. An example here is BWE, and this is what the IGSN looks like, the example IGSN. And then you can batch upload your sample metadata file. As Stephanie asked that question, you just include all of your samples in a file for a given sample type. You can update your metadata as needed and also add links to related data. And I'm going to go through a demo of that now. So here is the CSAR website, and you just click on register samples. You sign up to be a GeoPass user. It's just a very simple form providing your name and email. And then, so I'm just going to sign in here. And this is also when you sign in, you provide your user code that I mentioned, that 3-character code. So this 3-character code, again, it's usually individual or project specific, and you can continue to use it over time. And then to upload your samples, you click on this upload my batch samples, and then you add a file. And I'm going to add a file from our folder here, the completed IGSN example. And then you click upload, and it'll give you any errors. And then if there are no errors, there are errors here because I think I may have, let me see here. These IGSNs are already registered. And then after you submit your samples, you will usually get your IGSNs within an hour. And this process has recently become more efficient. Sometimes it would take a day or two, but now they usually get back to you within a few hours if you do it within business hours. You can also update your existing samples using this update link, and it's the same process. You add your file, update IGS. I'm going to just use this updated IGSN related by adding the related URLs for related data sets. And then you click upload, and it'll go through this and give you any errors that may come up. So that's how it'll then give you a view of what you've just uploaded. You can also click on these tabs above. There's a My Samples tab. And you can view all, it's taking a little bit of time because I clicked on this update, and sometimes the updates take a little bit of time, just like a few minutes. So yeah, when you click on My Samples, you can select one of the useful things that some of our pilot test participants found useful is the label printing. So you can select any of your samples. These are just all of the samples that I have registered, and you can use their tool to generate labels, label templates, and they have 2 different sizes here. If you need additional sizes, I do have another useful tool to generate labels, so you can contact us if you want a different way to create your labels. And it also has a barcode. So it has the IGSN and your sample name and a barcode, which can be very useful for your sample tracking. And so the final step is to publish your sample data on ESS-DIVE. And so that your data sets for your sample data sets in ESS-DIVE should include all of your sample related files, including your sample metadata and any of your sample data after the analyses have been completed and you have those available. One thing that's very important is to include is to make sure that you include all of your IGSNs and your sample names within all of the files that contain sample data. And this is how we can associate the samples and all of the metadata that go along with those samples, like how they were collected and their location, with the subsequent data. After you publish your data on ESS-DIVE, you can then update your CSAR IGSN sample metadata with related URLs. And there's a file in the sample tutorial folder that shows you how to do that. I'm going to skip the last poll questions because we're running out of time and just go over the conclusions here. So I hope that through this tutorial, you now have the resources to help start with your sample data management, including planning out how you want to manage samples and related data, including file organization and assigning unique identifiers, how to standardize metadata using ESS-DIVE spreadsheet template, and then how to register and manage your samples using CSAR when you want to register your samples for IGSNs. And so again, the overall purpose of IGSNs is to address this sample tracking problem that's been prevalent in the community so that you can more effectively track and exchange information about samples over time and across data systems, publications, and collaborators. And so this has really been especially important for collaborative and interdisciplinary environmental system science projects. And so I can take just a few minutes for questions. I can stay on a few minutes after if you would like to ask some additional questions after the hour, but I'll open it up now for any questions. And let me look at chat as well. So Terry asks, once the IGSN is assigned in the uploaded spreadsheet, do we download the file from IGSN to include the submission to ESS-DIVE? They will actually send you an email. You can download the file from the CSAR system. They'll also send you an email with the file that includes the IGSN. So the CSAR will, you provide your unique sample names and all of your metadata, and then CSAR generates the IGSNs, and they will send that to you along with, in a file along with all of your metadata.
I think the question was, how does it then get into ESS-DIVE? You upload it.
Yeah,
with
and
that.
then so you download it. Yeah, and you include that sample metadata file in your ESS-DIVE data set as well. Some people are even publishing a data set for just the sample metadata. And then they have a separate data set for different data analyses that are conducted on that set of samples. But I think the most common way to do it would be to upload your sample metadata file along with all of the data files that go along with those samples in your data set. Okay, thanks everyone for joining this webinar. I'm going to stop recording now and